So I think probably the major challenge for the next 10 years is working out how we get humanity to stop putting so much carbon into the atmosphere. We're completely addicted to energy, and that encompasses all our lives, sometimes in ways that we don't really think about. So, for example, you may or may not realize, but the UK uses 5% of all the electricity it consumes to make drinking water. So I'm not going to talk about drinking water. As you can probably tell from the title of my talk, I'm going to talk about this. So what you can see here is wastewater. This is a small fraction of the wastewater. This is actually at the water treatment works down the road in Neyburn, where they treat the equivalent of about water from 250,000 people. So Yorkshire Water produces about 2 billion litres of drinking water a day. Obviously, most of that turns into wastewater. You can see here from the colour that there's stuff in that wastewater. That's mainly organic material, so it's been biologically produced. And we need to do something with that waste. So what actually happens in water treatment in the UK is the raw water comes onto site. It goes into large funnel-shaped containers, and most of the organic material is allowed to settle out, and that's called primary sludge. Then the dirty water from this process is decanted out into a secondary vessel, like the one I just showed you, and we blow bubbles into it. And those bubbles allow microbes to grow. Those microbes grow on the organics in the water, the waste, turn it into more microbes, and then settle out so that the water is cleaner again. But again, even in wastewater, that uses 1% of all the UK's electricity. And this was a good idea at the time, but this is essentially Victorian technology that we're talking about for treating wastewater. Then the cleaner water now is decanted again into trickle bed filters. You've probably seen these on the sides of motorways. And again, there's another biological process going on here that really polishes the water so that what's discharged as final effluent back into the rivers is cleaner than the water that's taken out of the rivers to make drinking water from in the first place. Yorkshire Water assure me that when everything's working properly, you can drink this final effluent, but I've never managed to find anyone that will actually do that in front of me. So the bit that I'm really interested in is the primary and secondary sludges that are collected here. They're put into another vessel, which is called an anaerobic digester, where some very ancient, complex microbiology is going on. That's done in the absence of oxygen, hence the anaerobic. And it is a very complicated microbial community. And by doing that, that organic material is converted into biogas, which is a mixture of methane and carbon dioxide, and nitrogen-rich digestate, which can be used as fertilizer. Now, this microbial community is not an artificial community. It exists in nature. It's been around for a very, very long time, and it happens in the environment. So, for example, anywhere where there's an absence of oxygen, you'll get this kind of microbial community forming. Lakes are a good example. The University of York has a lake, and sure enough, if you stand there for long enough, you'll see bubbles rise to the surface. If you collect those bubbles, you'll see that they're biogas, and the easy way to demonstrate that is to set fire to it because it's methane. So if you take your son into the lake, don't know why this is, okay, then you can collect the gas and set fire to it. So again, clearly, there's a biological process here that we can use to break down the waste that's in our sewage, but we can't just dump all our sewage into water bodies like rivers and lakes. I mean, that is something that we've done in the past. That is something that still happens in certain countries. I won't name any, but I have been places where that is the case. So instead, what we do is we engineer this whole process. And this is Yorkshire Water's most recently commissioned plant, Nostrop in Leeds. This is a £75 million investment in treating sewage sludge. And this hopefully gives you some sort of idea of the scale here. These are the anaerobic digesters that they've built at Leeds. Each of those digesters is 8 stories high. They stand 22 meters from the ground up. Each one of those contains 7.2 million liters of sludge. And it produces a lot of biogas, some of which is held in this gas bag here until it can be converted into electricity. So this is great, but this is £75 million worth of investment, basically, to treat the sludge in Leeds. So what happens as the population grows? Obviously, you can build more and more facilities, but that gets expensive very, very quickly. So the goal of the work that I've been doing, which has been sponsored through the Royal Society and with Yorkshire Water, is to see if we can improve this process. There's been a lot of engineering around moving sewage and mixing sewage, but there's been very little involved in engineering the microbial process here. So can we basically put more material through the same assets so that no more need to be built? And that's important from a carbon point of view, not just from the energy point of view, but because, again, concrete is a huge emitter of carbon dioxide. So actually, concrete produces about 8% of all the carbon dioxide emissions that we have globally. So we don't want to build more things. So the problem is, if you're dealing with facilities at 7.5 million litre scale, then people don't want you to do experiments with them. And you know, the infrastructure for sewage sludge needs to work 24/7, 365 days a year. You know, if it breaks, you would know, and there would be a lot of very unhappy people. So to get round this, again, Yorkshire Water have invested in facilities at the University of York, which is shown here. We now have 60 5-litre anaerobic digesters in the biology department, which allow us to model the process that we're seeing at a very large scale and try and optimize without interrupting normal services. And clearly at the center of this is microbiology. So here's one of the key microbes that has been grown in my lab, which actually produces methane at the end of this process. And there are a lot of individual microbes in these communities. Okay. So for each milliliter of sewage sludge, we're talking about 100 million microbes. Okay. So that means that we've got billions in each of our 5-litre pots. And again, if you go back to the really large-scale systems, then this is where you realize you should check your facts before you produce your slides. So one of my colleagues said with great authority that there are more microbes in an anaerobic digester than there are stars in the universe, which, you know, he's a very eminent scientist, so I absolutely believed it. Produced this slide to say, in an anaerobic digester, there are more microbes than there are stars in the universe. And then I thought, you know what, I better check this. So I went to the European Space Agency, and they say, they estimate there are between 10 to the 22 and 10 to the 24 stars in the universe. It's obviously only an estimate. We can't count that many. I did a calculation, and I realized that we're a little bit off. So, but surprisingly, not very much. So if you take these 4 digesters at Nostrop, they have somewhere between 1/3 and 30 times fewer cells than there are in the entire universe in terms of stars. So if you look actually at anaerobic digesters across the UK, yeah, there are more microbes in anaerobic digesters than there are stars in the universe. So this is a complex system with a lot of things going on. How do we understand this? How do we try and make sense of it so that we can start to engineer this community? So what we've done is we have gone not to this facility, because this facility has only been online for about 6 months, but a similar facility at Neyburn in York, just by the designer outlets, and sampled the microbes over a period of time. So Neyburn also has 8 digesters. They're not quite this large. They're only 1.8 million litres each. We collected a lot of samples over 8 or 9 months, and we started to analyze the DNA. Now, traditionally, DNA sequencers are enormous and expensive. They're kind of the size of washing machines. They take a long time to run. They give you a huge amount of data, which is difficult to analyze. But things have changed quite dramatically recently. So now, the latest DNA sequencers, and this is an instrument that's produced by a company called Oxford Nanopore, which is run actually by a York biology graduate, and you literally can put your DNA sequencer in your pocket. Now, this is good for a number of reasons, not just because it gives you something to wave around when you're giving this kind of talk, but it means that sequencing is cheaper, it's also faster, and it's giving us a different sort of data. It's giving us much longer pieces of information, which theoretically are easier to analyze. In reality, that's not the case. So we started sequencing using this technology and then realized that we had an issue with analyzing our data. So we initially used York's supercomputer. At the time, we had a facility called York, which turned out to not be very super. We uploaded data onto it. It ran for a month, and it crashed, and we didn't get any results. Fortunately, the University of York has a collaboration with Google. They allowed us to use Google Cloud to do this. After 2 weeks, we did get a result. They told us it would have cost £8,000 were we to do that usually. But in the meantime, the University of York had invested in a much more supercomputer, which is called Viking. We managed to sneak on it as soon as it was switched on and before the rest of the university was using it, and managed to analyze our data in just over a day. And this is the kind of thing you see. So each one of these panels now is an individual microbe that we've been able to identify from within that mixture of microbes living in the community in the anaerobic digester. And if we zoom in on one of these panels, you can see that this is how this particular microbe has behaved over 9 months in digester 1, 2, 3, and 4. What's remarkable here is that even though these are operating at enormous scale, the community within these individual digesters is reacting in the same way to the material that's going into the digester. And what that means is this microbial community is ultimately very predictable, which means that we really do have the potential to engineer this. And this is the first time it's been shown at any kind of scale that these microbial communities, despite having thousands of different species and despite having trillions of different individuals, are behaving in a very predictable way. So what does that mean for us? I'm thinking my slide should... Oh, no, that's not what should have happened. Can we go... oh, okay. Right. So, anaerobic digestion is a great way of recovering resources from sewage. And in fact, now those are called bioresources. Obviously, we get biogas from this, and that can be used to create electricity and heat. We get digestate, which can be used as fertilizer. But AD doesn't just work on wastewater material. It works on all sorts of organic waste. So food waste is another thing that can be used in anaerobic digestion, and importantly, farm residues. So the statistics are, if we were able to collect all of the animal manures produced by agriculture across the world and put it through anaerobic digestion, that would generate enough power or energy for all of global agriculture, and we'd still have energy left over. There are more things we can do with this. The biogas can be used just for domestic cooking as a drop-in replacement for natural gas, but equally, it can be upgraded to biomethane and used to run lorries. And it's really, at the moment, the only viable alternative to fossil fuels is compressed methane in these kinds of trucks. It can also be upgraded and put directly into the grid. And if you live in Sherburn in Elmet or other places locally, a proportion of the gas that you're using from your mains is biogas. But we can go further than that when we start to engineer the community. We can reform the biogas into methanol and hydrogen to make even cleaner fuels. but we can also engineer this community so that it produces more interesting molecules straight away rather than methane. And there's already been some very nice work done in the States where they've demonstrated the production of isoprene, which can be directly used as a petrol replacement, or we can start to engineer higher value molecules like the ones that Mike was just talking about. And at that point, I need to stop, say thank you to all these people, particularly the Royal Society and Yorkshire Water for support. Thank you.